According to the paper, the brain consumes 20% of the body's total energy. It is evident that the brain needs constant energy supply, meaning it must be continually fed with oxygen and glucose taken from different locations. If these requirements are not met, then there will be a reduction in cerebral blood flow, which results in cognitive impairment or even damage to tissues. Alzheimer's has a correlation of reduced cerebral blood flow to the cortex. It also exhibits a correlation with cerebrovascular diseases. This could essentially cause even further neuronal damage, so neurovascular function as a whole may contribute to the impact of Alzheimer's and its damage on patients. Amyloid beta is a pathological hallmark for Alzheimer's. These deposits precede cognitive decline by multiple years, proving how dangerous it is and its connection to Alzheimer's. In a typical brain, glial, neural, and vascular cells work together as a function system called a GVU or gliovascular unit, which effectively ensures greater blood flow with greater neural activity. This connection between greater neural activity and greater blood flow is referred to as functional hyperemia or neurovascular coupling. Astrocytes are placed within these gliovascular units to aid in the function of hyperemia. There are two processes within the astrocytes. The first is the large perivascular end feet that coat the cerebral vasculature, while the second is the finer processes that enwrap the neuronal synapses. These astrocytes respond to activity by increases in intracellular calcium, which causes the release of factors that act on contraction-inducing cells within the gliovascular unit, known as vascular smooth muscle cells, VSMC. When Alzheimer's disease is present, gliovascular unit anatomy is altered. Studies have shown changes like endothelial cell degeneration, blood-brain barrier abnormalities, and even capillary distortions.
The rationale for this experiment is to relate gliovascular unit functioning to shed light on its relationship to Alzheimer's disease and to question if cerebral blood flow to the gliovascular unit, GVU, has any relationship to Alzheimer's. It was hypothesized that vascular amyloid may physically interfere with gliovascular signaling, and this may contribute to cerebral hypoperfusion in Alzheimer's disease.
Mice were the test subject in these experiments. Hemozygote males were bred with females to produce HAPPJ20 mice. For the electron microscopy, test subjects and control brains were transcardially perfused with PBS. After fixation, the brains were removed from solution and sectioned into 500 to 1,000 micrometer slices. Samples were washed in 3 different buffer washes in differing environmental conditions to maintain pH of 7.4. Ultra-thin sections were then cut and imaged on a microscope. Tissue of HAPPJ20 and control mice were fixed in paraformaldehyde in PBS, then coronal slices were taken. The slices were incubated in primary antibody solution overnight, washed in PBS, then stained with secondary antibodies. Then a DAPI stain was used. Confocal images were taken for each genotype and time point in the cortical gray matter of 3 different tissue slices to quantify GFAP and AQ4. Mice were sacrificed and their brains were placed in ice-cold artificial CSF. 200 coronal microtome slices were sectioned. For TACPD experiments, vessels were pre-constricted for 30 minutes in recording artificial CSF. The brain slices were observed under fluorescent microscopes, and the vessel diameter was measured as the cross-section of the vessel. Calcium uncaging was achieved using a 405 nanometer laser. The effect of laser scanning on caged calcium was assessed by the vessel diameter and changes in the presence and absence of the high-powered uncaging pulse. A cranial window was added to one hemisphere after the skull was removed. To visualize amyloid, methoxy X04 was injected. Arterioles were identified by injecting hydrazide.
Methoxy X04 was injected into the body cavity before in vivo imaging by multiphoton microscopy to visualize vascular amyloid in the brains of aged HAPPJ20 mice. As Alzheimer's progresses, gaps in vessels shrink and vascular amyloid starts to form continuous structures. Based on high-resolution confocals, vascular amyloid had a banding pattern that resembled an exoskeleton around blood vessels. 3D reconstruction data showed vascular amyloid volume to be approximately 20% or the total vessel volume. This image displays blood vessels labeled in pink that are coated in amyloids labeled in green. These amyloids provide structure but can also result in constriction of the vessels or in the inability for dilation of vessels. In this experiment, preferential loading of astrocytes by fluorescent acetoxymethyl ester indicators were used to label and visualize astrocyte end feet. Hydrazide dye was used to stain the elastin layer of arterial vessels. The astrocyte-specific marker GFAP was used to compare the extent of astrogliosis at different ages in HAPPJ20 mice. In 3 to 9 months, GFAP expression is comparable to age-matched littermate controls. At 12 to 16 months of age, an increase in reactive astrocytes around amyloid plaques previously reported in a different Alzheimer's disease mouse model was observed, but there was no significant difference between HAPPJ20 and the control. At 24 through 29 months of age, astrogliosis was widespread through the cortical gray matter at an appropriate level for age-related animals. Electron microscopy was used to assess structural relationships of astrocyte end feet and blood vessels in control of the mice, which is characterized by less electron-dense, lighter cytoplasm. Conversely, amyloid deposits are relatively darker structures close to the vessel wall and are not observed in control mice. As age increases, the area GFAP immunoreactivity increases significantly between the control and HAPPJ20 subjects in the left graph. In the right graph, as age increases, the area of AQ4 immunoreactivity is seen to decline from the control to the HAPPJ20 groups, declining to the point that the results are no longer significant between the control and the HAPPJ20 groups. Functional hyperemia is a process of brain regulating the blood flow in response to neuronal activity and energy demands. The activation of astrocytic metabotropic glutamate receptors lead to the influx of calcium and activation of second messengers that dictate arterial cell contraction and dilation. Vasodilation or vasoconstriction are dependent on an increase in calcium levels and oxygen tension in the tissues. In the left graph, TACPD was thought to result in the dilation of blood vessels, however, results generated no significant difference. In the right graph, norepinephrine was thought to result in constriction of blood vessels, however, results generated no significant differences. Methoxy-XA4 dye was used to identify vessels with or without vascular amyloid. Hydrazide was used to determine changes in vessel diameter. Calcium was purposefully raised in certain astrocytes to control effects of TACPF and norepinephrine. To determine if the impaired vasoconstriction in HAPPJ20 mice was due to the amyloid burden on the vasal culture, the vasoconstriction of the arterioles with and without vascular amyloid were compared. In HAPPJ20 mice, response was comparable to arterioles from control mice. Images D and E show the maximum and minimum vessel constrictions, showing reduced responses in HAPPJ20 only when amyloid was on the vascular wall. Image F shows the mean to ensure that the effects were not due to variability in calcium release. The mean calcium was found and no significant difference was noted, meaning that arterioles burdened by vascular amyloid have a diminished range of contractility.
Astrocytes regulating blood flow in correlation with neural activity is now well recognized. It was also confirmed by electron microscopy that amyloid is deposited between astrocytes and the blood vessel wall. In glioma, the vessel diameter changing substances released by astrocytes were unable to reach the arterial smooth muscle. This caused a question of if the vascular amyloidosis may similarly disrupt the GVU. It did by diminishing the ability to constrict after astrocyte stimulation by calcium release. If AQ4 is lost or end feet are impaired, clearance of amyloid via interstitial fluid is disrupted, leading to its buildup. By understanding that amyloids reduce cerebral blood flow, it is possible to conclude that the neuronal pathways are atrophied, which increases the effect of Alzheimer's. Critiques. The experimental procedure could have been more clear. More trials could have been performed. There is no mention of the sample size of mice. Future studies could also look at how specific cells are affected by the presence of amyloids. Additionally, the research found that AQ4 is mislocalized by the mice, so it might not accurately represent astrocyte labeling. Amyloid deposits are mainly responsible for the impairment of the vasculature, which leads to the question, can the dissolution of vascular amyloid restore normal vessel function? This study suggests that there is a time window where therapy can result in amyloid clearance to allow the vessel to regain its full dynamic range. This therapy can then be developed for future Alzheimer's patients.